So as we said, the sugar may sometimes need to cross the plasma membrane, and as we said, that may happen, and this is governed by the selective permeability of the plasma membrane. So the plasma membrane is characterized by being selectively permeable. It will allow some substances but not the others. Usually molecules that are hydrophobic in nature, they can cross, especially if they are small in size, they can cross the plasma membrane, while, as we know, sugars are not hydrophobic. That's why usually the transport of sugars will not take place through simple diffusion. So what the plant will usually do, the plant will usually generate a proton gradient. So as you can see here, what is happening inside the plant, we have proton ions, this H plus. So the plant is pumping more protons from inside to the outside. That will generate a proton gradient. So proton gradient is a type of electrochemical gradient. So outside of the cell we have more protons than inside the cell, and to keep pumping protons from inside to outside which is against the concentration gradient, this needs investment by spending energy in the form of ATP. So the plant will spend energy to pump protons from the inside of the cell, which is the low concentration, to the outside of the cell, which is the present as higher concentration. So what is the reason for that? Because this generated gradient, this electrochemical gradient or this proton gradient, will be used later on as a source of energy to move other molecules like the sugars, the sucrose we are interested in, to move sucrose against their concentration gradients via co-transporters. So this will be shown in next slide. So if you remember from previous slide, the plants have pumped protons from inside to outside that generated electrochemical gradient or proton gradient. Now this proton gradient will be used as a source of energy to pump other molecules, which is sucrose, to pump other molecules, because now this proton collected here can move back to inside the cell, and now from high concentration to low concentration, this doesn't need energy. So the energy that the plant invested in the first part is now being used to co-transport sucrose from the low concentration to the high concentration. Remember, inside these mesophils, we have very high concentration of sucrose inside the mesophils. So to keep pumping sucrose inside the mesophils and to pack them with sucrose, that need to be done in this way using co-transport mechanisms, and the energy will be provided by the proton gradient that the plant has generated. Okay, so moving on to the sugar conducting cells of the phloem. So this is a bit of anatomy to see the anatomy of the phloem in more details. So the phloem consists of the sieve-tube elements. So these are alive at functional maturity, but they lack the organelles, and we will see how they maintain to do that, how they lack organisms lack organelles, but they are still considered alive. And the reason for being lacking these organelles, so let's first highlight the sieve-tube elements. So these are the sieve-tube elements. So as the name implies, it's like tube, and we have like many of these cells connecting end to end to like one another. So like this is cell number 1, this is cell number 2, and so on. So these tubes stack on top of one another, and they are connected through this sieve plate, which we'll discuss later on. So as you can see, the main function of the sieve-tube elements is to transport the aqueous solution, which is rich in sucrose, Remember, we said that it is the phloem sap. So in order to meet the function, it is better for this phloem to be composed of like tubes. These tubes are empty in order to transport more and more of the phloem sap easily. So that's the reason for lacking the organelles. Then they are connected through sieve plates. So these are the porous end walls that allow the fluid to flow between the cells, between one sieve-tube element and another sieve-tube element. So you can see it here, this sieve plate, this porous structure here and there. So this allows that transport to continue, and it is shown here, this black or dotted dots. Okay, so each sieve-tube element has a companion cell. This companion cell has a nucleus and ribosomes, and you can see it here. This is the nucleus of the companion cell, and those nucleus and ribosomes, they serve both cells. They serve the companion cell and the sieve-tube elements. That's why they are considered alive, because the organelles are actually being utilized from the companion cell. Okay, so now moving on to the long distance transport, and here I want to highlight one word we mentioned before. So the short distance transport, we mean by short distance transport moving from one cell to another cell, so transport between one cell to another cell, while long distance transport means a movement throughout the whole plant organism. So moving out through the whole body of the plant, different organs of the plant. So this is the long distance transport. So long distance transport, and here we are highlighting the role of bulk flow. So efficient long distance transport throughout the body of the plant. So this transport of fluid requires bulk flow. What does bulk flow mean? Bulk flow means the movement of a fluid driven by a pressure. So in order to transport fluids throughout the body of the plant, this needs to be done under pressure, and this is done through the movement through xylem and phloem. So water and solutes will move together. So this is important. So this requires the movement of water and solids together, and this happens through both xylem and phloem. So the movement through the xylem, tracheids and vessel elements, this will be covered in a next video. So here we are going to continue again our focus on phloem and the transport of sugars and the role of the sieve-tube elements, which we covered before. So let's summarize nearly all what we have mentioned in this video. We will summarize it in this slide. We'll put everything together. So we are going to discuss the bulk flow by positive pressure, the mechanism of translocation. So do you remember, translocation is the movement of the products of photosynthesis through the phloem. So what will happen? If you remember, the sugars or sucrose, the products of photosynthesis, they are produced in the mesophyll. So this orange cell is the mesophyll. It is the source or it is the producer of the sucrose. So the first step is the transfer or the transport of sucrose from the mesophyll to the sieve-tube element. So the first step is to load the sugar into the sieve-tube element, and this requires co-transport, as we said before, with the protons. To invest the proton gradient in this loading, and this movement here can happen through the two pathways which we have already mentioned, the symplast and the apoplast. Okay, now the sieve-tube element is being loaded with sucrose. What will happen next? What will happen next is the uptake of water. So this water will go through the sieve-tube element, either through the xylem vessels or through the mesophyll cells. So water will move through the sieve-tube elements by osmosis, and remember the xylem is close to the phloem, so the osmosis can work moving the water from the xylem to the phloem, or it can even come from the mesophyll cells themselves. Anyway, at the end, now we have sucrose and we have water, all of them inside the sieve-tube elements. Since the sieve-tube elements are composed of rigid cell walls and they are not going to extend, so the rigid cell walls will stay in place, this will generate a positive pressure. This will generate a positive pressure pushing the water, and now it is carrying loads of sucrose, so pushing this phloem sap through the cells of the sieve-tube elements till they reach the sink. So once they reach the sink, for example the root cells or any non-photosynthetic cell, the sugar will start to be unloaded at these sink cells. This unloaded is facilitated by facilitated diffusion. So this unloading is done through the facilitated diffusion, and we are going to see this in the next slide. So the sucrose is going to move from the sieve-tube element to the sink cell through facilitated diffusion. What will happen next? Water is going to be recycled back to the xylem like that in the fourth step. So this summarizes the whole mechanism of translocation, and to finish this video I will just zoom in to show you this step and how facilitated diffusion is used to transport the sucrose to the sink cell. So as we mentioned, sucrose is sugar. It is not a hydrophobic molecule. It cannot pass through the membranes by simple diffusion. So what do we need is facilitated diffusion. Why it is diffusion? Because it is diffusion from the high concentration gradient from inside the sieve-tube elements to a low concentration gradient at the sink cell. Why it is facilitated? Because it happens through a carrier protein. So there is a channel protein that this channel protein is allowing the passage of sucrose from high concentration to low concentration, so it is a facilitated diffusion. That's the end of this video. Thank you so much. Please stay safe and stay healthy. Thank you.